All right, it is 1 o'clock, so we will go ahead and get started promptly. Thank you very much for coming to the very last Science Division pop-up lecture of the year. My name is Paula Cushing. I am curator of invertebrate zoology here at the museum, and it's my pleasure to talk about these animals today. These are called camel spiders, solifuges, and I am going to give you a 25-minute overview of the last 24 years of research my lab has been doing on these animals. So tighten your belt because it's going to be quite the ride. So what are these animals? These are animals. They're in the kingdom Animalia. They're in the phylum Arthropoda, class Arachnida, and within that class, there are a whole bunch of different orders. So spiders are in their own order. We are not talking about spiders today. Just forget about spiders. They have nothing to do with today's lecture. These guys are in the order Solifugi, and during the course of today's lecture, I will refer to them as solifugids or camel spiders interchangeably. But just know that that's a misnomer. Now, when I arrived at the museum in 1998, I've been here for 25 years, I had never published a paper on solifugids. I didn't know very much about them. I didn't start doing research on these animals until I arrived. And the reason I started doing research on these animals is all because of the influence of this gentleman who's in the audience today, Jack Brookhard. So Jack was, career-wise, was a science teacher. He was a biology teacher in the Cherry Creek High School District, and also a football coach. I'm not sure which one took precedence in his life, but he was a science nerd. And this was back in the mid-1960s when Sputnik had happened and when the government was providing a lot of incentive programs to encourage science teachers and others to get higher level degrees, to get masters or PhDs. And so Jack wanted to take advantage of that opportunity. Well, one day he was out, he told me, and he was flipping rocks in the field, and he found a solifugid. He found a camel spider. Bizarre-looking animal, didn't know much about them, so he researched them. He found out what they were, and he thought to himself, well, this might be something intriguing to look at for my master's degree. So he figured out there were only 2 researchers in the world who did any kind of research on camel spiders. One of them was this gentleman on the left, Dr. Martin Mumma. And so Jack wrote to these researchers, and he asked about solifugids, tried to get some more information about them. And they both responded, and Martin wrote him back and said, well, you know, there's not very much known about these animals at all, but I encourage you to study them. So Jack very cleverly thought to himself, if I do my master's degree on a group of animals that nobody knows very much about, my committee members can't ask me any hard questions. So that was one reason he decided to pursue his master's degree and study these animals. So he started collaborating. Martin Mumma was his mentor, his advisor for his master's, and Jack started doing research with Martin and continued that collaboration until Martin's death in the late 1980s. But that didn't stop Jack. He continued his research on these animals, collaborating with different people throughout the country, looking at these camel spiders. And in 1998, when I got my job here, I found out that Jack was in the area, was in Denver, and I had heard his name. I had read papers by Brookhard and Mumma and Brookhard. And Jack found out there was an arachnologist at the museum, and neither one of us can remember who reached out first, but one way or another, Jack started working in my lab. And this was way back before we even had an arachnology collection. So Jack started working in the arachnology collection, and he started working with me, and he very gently pulled me in so before I knew what was happening, I was doing research on solifugids. They're a very difficult group to study, but Jack was, convinced me that I should do that. And so he and I started collaborating on the research on these animals. And in 2007, a colleague at the American Museum and I, Lorenzo Prandini, shown here on the upper left right there, he and I got a 5-year National Science Foundation grant, a big, over $1 million grant, to study the worldwide distribution of solifugids. And in 2018, another colleague and I, Matthew Graham at Eastern Connecticut State University, got another big, over $1 million NSF grant to study one family of sulfugids. Now, the overarching goal of both of these programs was, of course, to better understand the biology of this understudied group of arachnids. But another really major goal was to train early career scientists in this field. We wanted to make sure that it wasn't just me and Jack doing research on this group, that we could train the next generation of scientists. And these grants and these programs, with Jack's help, have been spectacularly successful. So we have had innumerable high school students through our Teen Science Scholar program come into our lab and get training, and through other internship programs. We have had multiple undergraduate students come in and get training from all over the world, as well as graduate students from all over the world get training in this group of animals. And these students are going on to continue this research. So we have succeeded in establishing a brand new burgeoning field of research in camel spiders. Now, when students first come in, these are just a few of the students. This is in the middle here is a former PhD student of mine, Tarina Bird, and another former PhD student, Dr. Erica Garcia, who is now pursuing her postdoctoral program in Old World sulfugids in Israel. Not the best place to be now, but that's where she is. Garan Shikak is in the audience. He's my master's student. Ryan Jones is my current PhD student. Another former PhD student who worked and got training in our lab, Anya Klan. Several Mexican students who my collaborators mentor for, this is Edmundo Gonzalez Santillan, came into my lab to get training. So we have done just a spectacularly great job in getting people excited about this group of animals. So what are sulfugids? Sulfugids are major predators of insects. They're voracious, pugnacious predators. They will eat anything they can. I think they would eat small children if they were large enough to do so. They are found in desert ecosystems throughout the world, desert habitats. So during the course of our research in these last 2 decades, we have done research in all the major deserts in North America, Sonora and Mojave, Chihuahua and Great Basin. We've gone down to Mexico to collect and to study these animals. They are nocturnal predators. So in the daytime, they're hiding in burrows, and I'll show you this video by Erica showing a burrowing sulfugid. But they come out, most of them come out in the evening or at night. They're nocturnal, and they start running, and they don't stop running. They will run for miles, looking for prey, looking for anything they can eat, looking for each other. And they are active pretty much from an hour after sunset until about an hour before sunrise. Running around out there. So in this video, you'll see a female, and when the sun begins to come up or conditions change, they will burrow probably wherever they are, and they'll just dig down. The females will probably establish burrows and stay in the burrows and protect their eggs. So this video, oops, I knew I was going to do that, will show a female that is digging her burrow. So she's going to shove that soil out with her chelicerae, her jaws, and her pedipalps and her front legs to establish that burrow. So that's what they do during the daytime. And at night, they come out of their burrows and they start hunting. Within that order Solifugi, there are 12 different families in that order. And in North America, there are 2 of those families, Amitrechidae and Aremabatidae. Our lab has been focused on the biology, the systematics, the phylogeny, the taxonomy of the family Aremabatidae, one of the more species diverse families, which has probably between 150 and almost 200 species in it. So what have we done in these last 20 years? We've done a little bit of everything. So one of the major foci of our research has been to better understand the relationships between and among the genera and the species within the family Aremabatidae. So remember back in your biology days, kingdom, phylum, class, order, family, genus, species, plural of genus is genera. So we've been using next generation sequencing and other molecular sequencing techniques to figure out the phylogenetic relationships, the evolutionary relationships between and among the 8 different genera that are found in the family Aremabatidae and the species within each of those genera. Now, once we have that backbone evolutionary tree that shows the relationships and shows which of the groups are monophyletic clades, or evolutionary lineages, well-supported lineages, then we can revise the taxonomy of the family. And that's what we're in the midst of doing right now. And in the process of doing that, we've also done a lot of research on their morphology, their anatomy. They have some really strange body parts and body structures. So we've been digging into some morphological studies of these animals as well. And then serendipitous other projects on their behavior, on their mating, on whatever comes up that is intriguing, that seems like an interesting question to pursue, we'll pursue it. So what I'm going to do now is just give you literally just the highlights, just a taste of some of the research that we've done over the years. So one of the really exciting products that's come out of our research in our lab, as well as other colleagues' labs, is this paper. This was published this year, and it came out of my colleague Prashant Sharma's lab in Wisconsin. The first author is one of his students, Siddharth Kulkarni, but you should see some names that you recognize in this list. My name, Erica's name, my student Ryan Jones, some colleagues of ours who contributed data for this study. And this is a major phylogenetic assessment of the entire order of Solifugi, asking whether the 12 different families within that order are good families, are monophyletic, are evolutionarily decent lineages. And it turns out that our family, Aremabatidae, is, and that some of the other families are good lineages. They're well supported by this DNA data, but a couple of the families are not. They're coming out on different points in the tree, which suggests this gives us a starting point to really dive in and figure out what's going on, figure out how evolution worked on this order. In 2015, my lab published with, of course, Jack is on this paper, published the first ever molecular phylogenetic analysis of this family. And if you've heard my lectures in the past, you've seen this tree. And in this case, we were using something called multi-locus Sanger sequencing. But again, it's a molecular approach to ask, in this case, are the 8 genera that are found within the family Aremabatidae, are those 8 genera monophyletic? And what we found, and you don't have to pay much attention except to notice how pretty the tree is with all those Christmas colors, but what we found out was that 5 of the genera are monophyletic, but 3 of them that include most of the previously described species, the really species diverse genera, are not. They're coming out all over the tree. So this kind of data gives us, and this kind of publication of an evolutionary hypothesis gives you really good starting point for asking the next question. And that's what our lab is doing now, using next generation sequencing, using more taxa sampling, more species. Then we've also touched on, I said, the morphology. We've done a lot of papers looking at the morphological characteristics of this group of animals. So in order to understand those studies, you have to know the bow plan. If Frank is in the audience, he would appreciate me using that word. The overall structure of the body of a sulfugid. So sulfugids, like a lot of arachnids, have 2 major body parts. The head region is called the propeltidium. Second body part is called the abdomen. They have 4 pairs of legs. They have a front pair of appendages called the pedipalps. When you see these moving in the environment, it's like you're looking at a little 8-legged zombie, because they hold their pedipalps aloft as they're running around the environment. The pedipalps are covered with sensory hair-like setae. So they use those pedipalps like insects use their antennae. And they also have this pair of super powerful jaws called chelicerae. So we have done some research on different anatomical, different morphological traits to do with several of these different body parts. We have looked and dived into the structure of the chelicerae. And for the chelicerae, I'm going to be talking about, they have an upper part of the jaw and a lower part of the jaw. The upper part of the jaw is called the fixed finger. And you just need to kind of remember that. Fixed finger and movable finger of the chelicerae. So we've done a lot of work on the chelicerae. Brian Jones did his master's work. He published a paper looking at the ctenidia. The ctenidia are these weird spiny-like structures that some male sulfugids have on the underside of their abdomen. We have no idea what their function is. Only the males have them. And so Ryan was looking at the phylogenetic signature. How useful are the pattern and the length and the number of tenidia in differentiating one group from another? Erica Garcia looked at the palpal spines on the palps of the sulfugid, because the spines have some interesting characteristics to do with them. We've really dived into the structure of the pedipalp as well, and some of the seeding on the pedipalp. So I'm going to be touching on some of these different studies that we did over the years. But let's look at the chelicerae first. I said that the upper part of the chelicerae is called the fixed finger, and the bottom part is the movable finger. What's interesting about sulfugid's chelicerae is that there's sexual dimorphism. The male chelicerae looks really different than the females. And in particular, look at the teeth. Females have teeth on the fixed finger and the movable finger. Males don't. And sometimes the male fixed finger is this crazy shape. And in some of the old world families, the male chelicerae has an additional structure called the flagellum. We have absolutely no idea what the function of the flagellum is, but it also takes on some crazy shapes. So you can see the diversity of these flagellum in the males of these sulfugids. And this is from the major publication that came out of a previous PhD student of mine, Tarina Bird, in 2015. So why do the males have these weird structures and weird morphological characteristics to do with their chelicerae? It's because they use their chelicerae to mate with the females. And those of you who have heard my lectures, you've seen the sex videos, you're going to see it again. So what you're looking at here is the male not attacking a female, but mating with her. So males, their testes, the male testes is in his abdomen. He has an opening in his abdomen to release the droplet of sperm. Female's ovaries are in her abdomen. She has an opening there to release her eggs. But males do not have a direct intromittent organ. They don't have a penis associated with their genital opening. And instead, what the male sulfugids will do is they will grab the sperm bundle, the little droplet of sperm, and they stick it into the female's operculum, her genital opening, with his chelicerae. It is very disturbing. So let me just warn you, because you're going to see a video of this, and it truly is, it'll give you nightmares, I guarantee. So this is a video by another student who got training in our lab. This was from a publication by Jen Roswell and myself in 2020. And Jen did her master's degree, very nice study looking at the mating behavior of sulfugids. And in this video, this animal here, that's the female, and she's running around. And you're going to see the male rushing at her in a moment. He's very excited. So here's the female moving around. And it's a mirrored terrarium, so you can see the structures underneath. Here's the male. Now what's interesting is as soon as he grabs her, as soon as he makes contact, she goes lethargic. She completely stops moving. She goes into a quiescent state. We have no idea what triggers that, but we think it's something to do with the contact by the male. Look at his pedipalps too. His pedipalps are holding onto her, and specifically he's using these little pop-out organs called suctoral organs to hold onto her abdomen and manipulate and maneuver it. And then what he's doing is he's sticking those fixed fingers, those, thank goodness they're toothless fixed fingers right into her genital opening. We don't know what's going on there, but we suspect it's either to prepare her for receiving his sperm or removing the sperm of competing males, or maybe both. This goes on for a while. And I don't know how else to describe it than that he chews on her. But we're going to skip ahead a little bit, because he'll do this chewing for quite a while, for several minutes. And then what you're going to eventually see is that he will remove his jaw from her genital opening. He will move his body upwards and directly deposit. He's about to pull his jaws out. He'll directly deposit right there. He's depositing the sperm packet right onto the surface of her genital opening. And then he chews again. Then he uses his fixed finger to push that sperm packet into her. And then he chews again, and that's probably to open up that sperm packet. This continues for another couple minutes. We're not going to watch it all. But what's interesting too here is that watch the female, she begins to wake up. So as soon as he's copulated with her, she begins to get more active. She comes out of that lethargic state. And then eventually she gets active enough that she can break away and run away. And if he doesn't run away fast enough, she'll cannibalize him. So Jen, in her study, demonstrated that the cannibalism rate is really quite high in these animals. So that was one study that we worked on with a graduate student. We also did this work on these suctoral organs. So these suctoral organs I already mentioned, they pop out, they're balloon-like structures, they can hold on to smooth surfaces. So they'll hold on and manipulate the female's body. Females also have these suctoral organs. This is a scanning electron micrograph of an everted suctoral organ. And both males and females use those suctoral organs to pull prey closer to their body as well. So we did this study using SEM, scanning electron microscopy, and light microscopy to understand the structure of these suctoral organs and how they worked. How were they able to pop out and get pulled back in? And it turns out that they become everted, and this was published in, there were two papers, one in 2005 and one that I was co-author on in 2008 with another student who got training in our lab. But it turns out that they will increase hemolymph or blood pressure into the pedipalp, and that causes the little balloon-like suctoral organ to pop out. And then they have tendons that will physically pull it back into the pedipalp when it's not in use. Another student and I, Patrick Casto, who worked in our lab for a while, did a nice little survey of the setae, of the hair-like setae that are covering the pedipalps of sulfugids. And in this study, we were trying to get at, are there any setae that are found in all 12 families across the order? And in fact, there are. You see these bifurcated setae in any family that you look at. They're very common. But there are some setae that we only see in a few of the families. In particular, these little Christmas tree. It's appropriate time to talk about papillae because it looks like a little Christmas tree. And these little papillae are found on the pedipalps of the males of 3 families. So we see those Christmas trees, and this is what they look like under light microscopy. This is the pedipalp here and the field of papillae. And we see these papillae in Aremabatidae, the family that we focus on, Cartiidae, a different family, and Sulpugidae. So we use various techniques to try to understand the structure and the function of these papillae. So we hypothesized that maybe only males have them, so maybe they have something to do with sexual selection or copulation or something. So we hypothesized that maybe they were mechanoreceptors. Maybe they could detect the movement of the female's body or his own body. Maybe they were picking up chemical signals from the air as olfactory receptors or contact chemoreceptors. Or maybe they had glands at the base and were spewing out some kind of a sexual signal that would attract the females. So we use various techniques to test these hypotheses, including transmission electron microscopy, doing sections and trying to look for dendrites, axons, nerves that would extend into the papillae. And in fact, we found some evidence that they were mechanoreceptors and chemoreceptors. We saw for mechanoreceptors, you would expect to see nerves at the bases, ending at the bases of the tree-like structure. This is a cross-section. And if it was a contact chemoreceptor, you'd expect to see dendrites or nerves that extend up into the shaft, and that's what we saw. We did not see any glands, so there's no evidence that there were secretory organs. But there is good evidence. This is what we think the structure looks like, with a dendrite that extends into the shaft, openings on these little branches, and tubular bodies, which are mechanoreceptive dendrites that end at the base. And we also used electrophysiology, where we took a little electrical probe on a live sulfugid and we trapped him. And then we put the probe right at the base of a papillae and exposed that papillae to different chemical cues, to water vapor, to temperature, to test whether it was picking up on any of those sensory signals. And that evidence suggested that it doesn't pick up on, we could reject the hypothesis that it was a thermoreceptor, reject the hypothesis it was a hygroreceptor picking up humidity. We could reject that it was a secretory organ. but we did get evidence that it was a mechanoreceptor and a chemoreceptor. So that paper was published in 2014. Erica Garcia, Dr. Erica Garcia, she did a study looking at the spines on the pedipalps of different genera within the family Aremabatidae to see if there were some spines that you only see in certain of the genera. And she got some evidence that there are some unique structures in some of these spines, these pedipalp spines. So this is in prep. Ryan Jones, my current PhD student, former master's student, did a nice study that was published in 2022 where he looked at whether these ctenidia, those weird structures on the abdomens of some males, whether they had any kind of diagnostic or phylogenetic signature, whether you could use those to figure out one genus from another, one group from another. And what he found is that the number has some signature, but not the shape or the length. And then we've really improved how we've collected these animals. So we've published some papers on how to maximize your collecting success. These animals are really hard to find, really hard to collect. They run like the wind. They don't stop running. But what's weird about them, and all of these times that I say it's weird, there are questions you could ask. I won't know the answer, but there are questions you could ask. So what's weird about these animals is at night they're attracted to light. They're positively phototactic. And because they're attracted to light, you can set pitfall traps underneath suspended lights in the desert. The lights will attract the sulfugids and they fall into the pitfall traps. So we've published a couple papers that have helped researchers worldwide maximize their success rate in collecting these animals. So, nearing the end, I just want to again stress how many students have come through our lab and gotten training, and how influential Jack has been on my own career. I would not have taken this step into this field of research if it wasn't for Jack. We have also produced products like this online field guide that have enabled researchers worldwide to better be able to identify these animals. So this field guide, we're publishing diagnostic characteristics that allow people to identify one species from another. We publish all of our data online and it's pushed out to the world. So the data that's available on sulfugid diversity is out there. This online database allows us to produce species distribution maps. And what's really exciting is that the field is burgeoning, the field is blossoming. So there are so many more papers coming out in recent years about these animals from all over the world. It is really an exciting time to be studying these animals. And it's such an exciting time that when I was at the International Congress of Arachnology in Uruguay, a group of us got together and I proposed the idea of writing a textbook, a revised textbook on the biology of these animals, because the only textbook that exists is obsolete, is outdated, and was never actually really very good to begin with. So they agreed, and we are well on our way towards writing a book proposal and the new textbook. The first draft, we're going to aim for 2026, 2027 to get the draft of the textbook done. And that all of this work would not have been possible and all of this excitement in the field if it weren't for Jack. So Jack has really spurred my career, and he has decided he's too old, he said, to come in anymore every week. So he and I are going to continue to collaborate, and I'll continue to work with Jack. Sorry, I knew this would happen. Anyway, I thank Jack because he has been incredibly influential in structuring my own career. So thank you very much. Garan, you want to? So I'm sure there are questions. I may not be able to answer them, but ask them anyway. I'm sure there are questions. Are there really no questions? Right, Jenna has a question. No, she doesn't.
I'm curious, how large do these creatures get, these spiders?
Yeah, so let me just, for the fun of it, show you this online photograph that I think people have seen. You guys have seen that? it makes them look like they're this big, right? But in the Middle East, you'll find the largest species in a family called Galeotidae. And it's the angle of the photograph that makes them look that big. That makes them look like they're 2 feet. The biggest ones are still large for arachnids. They're about 2.5, 3 inches in body length, and the leg length makes them almost small dinner plate sized. Cool story about, how many of you had seen this photograph before? Right, I met the photographer. So I was doing field work. A former student and I were driving through the Southwest and doing field work, and we had been camping in the deserts. We were hot, we were sweaty, we were tired. We wanted to stay in a motel on the way back to Denver. So we pulled off in some podunk town in New Mexico, the land of enchantment. And we checked into there was a night clerk at the motel. I mean, this was a mom and pop place because I'm too cheap to stay in named motels or hotels. So we go into this mom and pop motel, and there was a guy standing there, the night clerk, and he saw our government vehicle plates, and he said, "So, from Colorado, government from Colorado, huh? So what part of the government do you work with?" And I said, No. I said, "We're from the Denver Museum of Natural History," or at that time, Natural History. And he said, "Oh, okay, so what do you do there?" And I said, "Oh, well, I'm one of the curators on staff." He said, "Yeah, okay, so what do you do?" And I said, "Well, I'm a biologist." He's like, "Yeah, it's a natural history museum. I figured that. I mean, what do you study?" And he kept pushing it. And I said, "Well, I study arachnids." And he said, "Oh, like camel spiders." And I said, we have been on the road for 2.5 weeks, and you are the first person to know what a camel spider is. And he said, "Oh, well, you know that photograph," and he describes this photo. And I said, "Oh, yeah, I know that photo." And most people ask, do they really get this big? And so I jumped in and I said, "Oh, yeah, I know what photo you're talking about." I said, "They don't really get as big as they look in the photograph." I said, I think it's the angle of the photo and maybe the lens the photographer was using. And he leaned back and said, No. I took that photo." I was like, "Get out of here." And it turned out he was serving in Iraq, I think, and his buddies were fighting 2 sulfugids. One of them bit the abdomen of the other one, so one of his buddies stuck something down to break them apart, and the first one grabbed hold of whatever the object was, and they lifted them out of the terrarium, and guy's name is John Chandler. He shot this picture. He took this photo. So I left there. I was like, I cannot. It really is the land of enchantment. It's amazing.
Have you looked at molecular clock models since you're doing so much molecular analysis?
Yes.
Yeah.
So yes, have I looked at the molecular clocks in the 2015 paper and in the 2023 paper? We used a beast analysis approach in 2015 and molecular clock approach. And basically, in 2015, we have some problems with how they did it in the 2023 paper. But in our 2015 paper, my colleague Matt Graham, who's a biogeographer, used a beast analysis and basically demonstrated that as the desert environments were appearing, that's when you see this radiation of sulfugids. So it pretty much maps onto the appearance. The radiation of sulfugids matches the appearance of the desert ecosystems. So let's get this one, and then we'll pass the cube to you, Bob.
Sorry, you need the microphone box to speak, apparently. I'll pass it
Yeah,
away.
we're recording it, so.
Thank you so much for this, Dr. Cushing. I really learned a lot. I was always fascinated by how sulfugids are attracted to the light at night, and I am just curious to know if that protects them from possible predators or makes them more susceptible to predators at night, or if it has nothing at all to do with that.
It is such a
Thank
good
you.
question. And I have my own hypothesis. It's such a good question for desert nocturnal animals, is why they would be attracted to lights. If you think about it as an evolutionary biologist, it'll make you crazy. But the hypothesis that I have is that some of the research that I've read about moths and other night active insects that are attracted to lights is part of it is sky signals that allow them to fly or to track where they're going. But there was some paper that suggested that this light attraction in flying insects and in other groups of insects may have to do with an attraction to open spaces where it's easier to fly, it's a runway, it's an easier place to navigate. And so I wonder if maybe these night active predators like sulfugids have evolved an attraction to open spaces at night for similar reasons, except that maximizes their chances of finding prey. Is there support to that? I have no idea. And I would love to figure out some way to test why they are positively phototactic, because it's really intriguing. Now, Bob.
So they're worldwide. How do they move from continent to continent or from non-desert area? How do they move across non-desert areas? How does their biodispersal happen?
They have nothing limiting. To me, I don't think most of them have anything limiting their dispersal, because they have a metabolic rate more like insects, more like flying insects than like other arachnids. So their ability to move at night a very long distance is potentially substantial. Does that explain how they get from desert area to these separated desert areas? That's an excellent question that we're just beginning to scratch the surface on. So now that we have a higher level phylogeny of the order, then we can start asking questions about their biogeography, about those biogeographic patterns, and how the pattern of the families themselves may be explained by biogeographic patterns and the emergence of these habitats. Not a very good answer, but it's too early yet to answer that.
Okay, Dr. Cushing. We now know how you feel about Jack Brookhard. Can you tell us how you really feel about sulfugids?
Oh my gosh, look at the time. I've always had sort of a love-hate relationship with sulfugids, because when I say they're difficult to collect, they truly are challenging to collect at night. They run like the wind. One of the methods we use to collect them, because they move so fast, is we have a squirt bottle filled with alcohol and we shoot them with a stream of alcohol. We learned that from another colleague. So they're hard to collect. They're nearly impossible to keep alive in the lab for significant periods of time. Maybe a month, maybe 2 months is about it. I've had one female lay viable eggs in the lab, and those eggs hatched, and then they all died by the fourth instar. So they're almost impossible to raise or to rear in the lab. So my colleagues who have these lab cultures of hundreds of individuals of one species, when I publish my sulfugid papers, they're like, well, why didn't you use a bigger sample size? It's like, because we don't have a bigger sample size. So it's been a career changer for me to work on these animals. What I do like about them is they are fascinating. Pretty much any behavior or any pattern you see that has to do with their biology in the field is almost certainly something we don't know the answer to. So the number of research questions that you can pursue with these animals is endless. We are really just beginning. I mean, all those papers, if you really looked at the titles, they were mostly about taxonomy and systematics, not so much about behavior, not so much about ecology. So there are all of these really interesting questions about their basic natural history and biology that are yet TBD. And that's exciting. But the challenge of them drives me bananas sometimes.
Paula, is there any prehistoric or fossil records of these? I know they're soft and they don't really produce any fossils, but amber or any type of prehistoric?
Yeah, it's a good question. They're minimal, very minimal. There's amber. There's some amber fossils of sulfugids. And you're right that they don't fossilize very well because they're so soft. At least the abdomens are so soft-bodied. I have a sneaking suspicion that some trace fossils are probably associated with the jaws of sulfugids, but that's all you have, and so nobody recognizes them for that. And I don't offhand remember, do you remember, Garan, when the oldest fossil?
No, but I know, like, I think the most recent one discovered in the Baltic amber might be the oldest fossil discovered.
Yeah, so it's very recent fossils because they're almost all amber related.
Has anyone ever tried tracking these, like capture, marker, capture, or marking them with like radioactivity or something to see how far they disperse in one night?
Sorry,
or
we're
over
laughing
timeframe?
because Ryan tried that. So Ryan, he did a really nice study. The other paper that he published, Ryan Jones, my PhD student, for his master's, the other paper he did was a project he actually managed to carry out with his colleagues during the pandemic, where he went to Rocky Mountain Arsenal because we couldn't go very far, and he used fluorescent paint to mark all the individuals he could find, and then went night after night to see if he would recollect them, because we didn't even know if they had permanent burrows, if they would come back to their natal site, come back to a permanent site, or if you would mark them and then never see them again. And he found some evidence that they do have kind of a home territory, that they will come back to that little patch of habitat. So that's the only study that I'm aware of. Jack, do you know of any? Yeah, that's the only one that we're aware of where somebody's tried to mark and recapture them. We've talked about maybe using radio collars or finding out a way to use a radio marker. The problem with that is that they burrow, and we don't think the burrows are permanent burrows except maybe for the females. And so if you think about that, you know, I don't know what that would tell you, and you'd lose that signal until they come out again at night. So I think there's something that we could do there, but I just haven't figured out how. Other questions I can't answer?
Do we know what their like niche is in their environment? Like what do they do in their ecosystem that makes them so important?
They're major predators of arthropods in these desert ecosystems. So in these desert environments, probably the top arthropod predators, insect predators that you'll find are scorpions and sulfugids. And they've also been found in the guts of a lot of desert-adapted vertebrates. So not only are they top predators of arthropods, but they themselves serve as food source for other desert-adapted organisms. So they're an important component both from the bottom up and the top down of these ecosystems, of these desert communities. And if you think about deserts, I mean, they're pretty delicate environments. They're pretty fragile environments. They're at risk for degradation, irrigation, all sorts of habitat perturbations. And so the species that are adapted to living in deserts are not huge, you know. And so any community member is going to be important in maintaining that community structure in those really delicate environments. Other questions? Oh, one more.
Do you collect like tissue for DNA, like speciation identification? And if so, what part of the spider are you collecting?
Yeah, so what we'll do is we collect the whole animal because we have to use the morphology to identify it. And so we'll collect the whole animal. If we know we're going to use it for DNA analysis, we'll collect it not in 75%, but in 95%, higher percentage alcohol. And then what a lot of people will do is pull off a leg or two, and sometimes we'll do that, but we've also found that there's a lot of tissue in the jaw. And so you can tease that tissue out of the jaw and without damaging the exoskeleton and the structural integrity of the jaw itself. So we'll use the tissue, pull it right out of the jaw and use that tissue.
Thank you.
I think there was a question. Maybe, maybe not. Oh, okay. Other questions? All right, well, thank you very much. I appreciate it.